Hello students, I am Dinesh Udupi. Let us continue with our paper of molecular biology, evolution and ethology. This is session 6 and in this session we are going to study about PCR, that is polymerase chain reaction, and bioinformatics. Now what is PCR? Polymerase chain reaction. So PCR is a technique used in the lab to make millions of copies of a particular section of DNA. It was first developed in 1980s. The polymerase chain reaction was originally developed in 1983 by the American biochemist Carey Mullis, and he was awarded the Nobel Prize in Chemistry in 1993 for this pioneering work. So PCR is a technique. It uses a machine, of course, but basically it's a technique where we can multiply the copy of DNA. Say we have one or few copies of DNA and we want the same DNA in many number, like in thousands or millions of copies of the same DNA. Then we are going to use this particular technique called PCR, or polymerase chain reaction technique. Now, PCR is used in molecular biology to make many copies of small sections of DNA or a gene. Using PCR, it is possible to generate thousands to millions of copies of a particular section of DNA from a very small amount of DNA. And PCR is a common tool used in medical and biological research labs. So basically, PCR is a technique where we can obtain very high number of DNA molecules. So if we have a small fragment of DNA and we want it in large number, whatever the purpose, right? So in many fields, right, in biological research labs, in medical research labs, and also in the research of molecular biology. Now think of situations or think of conditions where you have a specimen, maybe a plant or animal or insect or whatever it is, and you want to study a particular gene or a particular part of the DNA. So you have a very small sample of DNA, but to analyze, to study the gene, you need a very high number of copies of those that DNA or gene. So in such cases, this polymerase chain reaction technique is used. Now, for this polymerase chain reaction, these are the requirements. So DNA template to be copied. Now this is the most important condition. So you must have a piece of DNA or the part of DNA sequence which has to be copied, right? So that is going to act like a template. And then primers. So you already understood in DNA replication topic that to create a new strand of DNA, primers are essential. So you must have a suitable primer. Now this is very important. You are having a piece of DNA or part of DNA or a gene, you must know the primer which can be used on that DNA template. So if the primer is not available, then PCR technique is not possible, right? So understanding primers and producing a suitable primer is essential in PCR technique. Then the nucleotide bases, these are the raw materials to produce the new DNA strands A, G, C and T in sufficient numbers. Then Taq polymerase enzyme. So this is the enzyme which is going to form the new strand, the polymerase enzyme. Then buffer to maintain correct conditions for the process. Since we are doing this in vitro, so outside the body in the laboratory conditions, it actually happens in a small machine, a PCR machine, which is a very small instrument. So you are supposed to maintain correct conditions, right? So that is possible by having this buffer. Now let us understand how the PCR process is done theoretically. So first there is denaturing when the double-stranded template DNA is heated to separate it into two single strands. Then there is annealing when the temperature is lower to enable the DNA primers to attach to the template DNA. And then extension when the temperature is raised and the new strand of DNA is made by the Taq polymerase enzyme. So just imagine what is happening in the PCR machine, right? So you have placed the buffer solution, which is going to provide the required condition for this multiplication or amplification process. So first what you need the DNA template. So that is your sample DNA. So theoretically you need only one copy, but normally you will be having many copies. So that is, those are the templates, right? So you know that that template DNA, the DNA molecule, the small fragment of DNA which you want to amplify is double stranded, right? So in this in this condition you have some you have other components also, right? So in the buffer you find the raw materials, that is nucleotides, Taq polymerase enzyme, right? And the buffer itself to maintain conditions. Now the theory is like this: when the temperature is increased, right? So it depends upon the uh, DNA which you are dealing with and the amount of ACGT concentration. So when the temperature is increased, you know that high temperature causes breakage of hydrogen bonds. So the two strands of the template DNA are going to get separated when the temperature is raised. Now once the DNA strands are separated, so the next step is attachment of primer to the strand, the template strand. So that is what we call it as annealing, right? So once the primer is attached, then the temperature is again adjusted so that the primer and Taq polymerase together start producing the complementary strand on the template strand. So in one cycle, you just imagine uh, you have only one copy of DNA as a template, right? So in the first cycle, what happens? The DNA, the fragment of DNA, the two strands get separated, right? And the primer gets attached to the template. So two templates are there. And then the Taq polymerase enzyme will start producing the opposite strand. So at the end of the cycle, you have two DNA fragments. So it is nothing but what is happening in the DNA replication. So DNA replication takes place inside the cell in vivo condition. But here the same thing is happening in vitro. So we are doing it in the laboratory. Again, the temperature cycle is changed, right? In the next cycle, the same is repeated. But only thing is, in the next cycle, there is doubling, say from 2 to 4. And after that, again, the cycle is repeated. Now we will be having 8 copies. So you are going to repeat this until you get the required number of DNA copies. Uh, unless and until all the conditions are met, so it is not going to happen. So you have to maintain all the proper conditions and you have to maintain sufficient number of raw materials, enzymes, etc. So at the end of many cycles we are going to get sufficient number of DNA copies, the fragments of the DNA. Now that's about the PCR. Normally what they do is after they get sufficient number of DNA to test the quality of DNA obtained, they will subject it to gel electrophoresis. So gel electrophoresis helps them to quantify and they will know, they will come to know that whether they got good quality or good number of DNA copies. And gel electrophoresis also helps to isolate, to separate the DNA which is multiplied or amplified. Okay, that's about the PCR technique. Next, let us move on to a new branch in biology, that is bioinformatics. So bioinformatics is defined as the application of tools of computation and analysis to the uh, capture and interpretation of biological data. Now, you can think of bioinformatics as a hybrid field where the computers are used to analyze the biological data, right? So here we are using very powerful computers, different kinds of softwares, and these computers and softwares are used to analyze the biological data, different types of biological data, right? So it is an interdisciplinary field which harnesses computer science, mathematics, physics, and biology. Now, bioinformatics, of course, nowadays it has developed to a very big extent, and there are so many things, there are so many theories, and there are so many practices which are made easy with the use of computers and softwares. And it is helping the biologists very much to solve many problems and to create new models and new ideas. Now, what we mean by biological data. So as far as bioinformatics is concerned, the biological data is nothing but the information about genes and information about proteins. For example, we studied the Human Genome Project in one of our previous sessions. So what happened in the Human Genome Project is they sequenced the chromosomes of humans. So all the human chromosomes were sequenced, and it created a very big data. So what we got is the sequence of nucleotides. The data is so huge, right? So we need computers to store it and to analyze it. Now, lot of information is hidden in that nucleotide sequence, and that has to be analyzed, right? So the first step is to store the data systematically. What is the data? In this case, the nucleotide sequence of the DNA. So that is stored first, and then it has to be obtained. So you have to recover, you have to take the data from that. So only the required data has to be taken, and then it has to be analyzed. So this obtaining data from the database, storing the data in the form of database, then getting only the required portion of data, and then analysis of that particular sequence data. So that constitutes bioinformatics. Nowadays, what is happening is there are so many different projects where the genome sequence, the DNA sequence of different organisms is done, and all that data is being stored, and where they are storing it. So the data is stored, and most of the data is available freely on the internet. Now there are some websites where we can get this data, and using different softwares we can analyze this data. So there are so many different fields nowadays which depend on this data which is stored in these particular websites. Now, like the nucleotide sequence of DNA, they also store the amino acid sequence of the proteins. So you know there are so many innumerable type of proteins in the nature, and those proteins are also analyzed, and their amino acid sequence is understood, and that amino acid sequence is stored in the form of database, and that database can also be analyzed for different purposes. So nowadays they are using different kinds of softwares to uh, design the three-dimensional model. So for example, the software is capable of creating the three-dimensional model, what we call it as the tertiary and quaternary structure of protein. If you give the amino acid sequence to the software, it will create the three-dimensional model. So it saves a lot of time and a lot of money in the drug design and many other such fields. So therefore, bioinformatics is a very important field of science nowadays, and its potential is literally infinite. So it's one of the fastest growing field in the biology nowadays. Following are some of the important websites where the gene sequences of many organisms are freely available. Now with the basic skills of computer knowledge, one can retrieve data and depending on the requirement of their work, they can use different softwares to analyze that particular data. Now, the first one is National Center for Biotechnology Information, that is NCBI website, uh, which is one of the most commonly used database in the bioinformatics. Then we have GenBank. Again, it is maintained by this NCBI only. It stores and archives DNA sequences from both large-scale genome projects and individual laboratories. European Bioinformatics Institute, ebi.ac.uk. So this is also one of the websites where you can get the genome sequences. Now, what is happening is there are hundreds of scientists which are involved in sequencing work. So they will uh, they will discover the sequence of organisms and then they will put it in these databases. So most of the databases are freely available, right? So all the scientists, they share their work. So anybody can access this data and can be utilized for their personal research. Okay, that's about this unit, this session where we studied the polymerase chain reaction, which is a technique to multiply, to amplify the DNA or gene of our interest. And then we just understood the concept of bioinformatics, which is a new field in the biology, which is a hybrid field using the computers, softwares to analyze the biological data. That is for this session. Thank you.